is a very exciting opportunity. Thank you very much to Marla and Lonnie and Shane for inviting me. It's a little daunting going up first, but here we go. I like to give my talks in a very informal setting. So if anyone has a question, shout out. Unfortunately, I can't see you. So you're going to have to shout out, or if you're too shy to shout out, tell Shane or Anya or Marla to shout out for you. And I'll happily answer any questions. So as Marla mentioned, I work at the South African Institute for Aquatic Biodiversity, and it's part of the NRF. And we deal with pretty much anything to do with aquatics. If it dips its toe in the water, that classifies. I'm going to give you, to start off the talk, just a brief background on what SIAB is and what we do, mainly because not a lot of people know about it, and we want to fix that. And also, if you are ever in Makanda and you want to come have a look at the collections and the lab facilities, etc., during Science Festival or any of those others, we do often have school tours coming through. So feel free to contact us and we'll happily facilitate that. So SIAB is got a range of different platforms. We've got everything from acoustic tracking array where fish are tagged and then they're monitored throughout the South African coastline. We've got the mapping, geophysics mapping platform, so mapping the benthic floor of the sea. Remote imaging platform, and that goes everything from coastal, shallow coastal, all the way to deep canyons. We've got 3 coastal crafts, big crafts that can go out to sea, as well as numerous smaller crafts that are used in freshwater research. Our national fish collection, it is the largest collection in Africa. So we've got the largest fish collection in Africa, and it's not just fish. We also have collections of cephalopods, amphibians, invertebrates, all sorts of things. And I don't have images here, but if you want to have a look at the collections, let me know. We also have the Aquatic Biobank, similar to what Marla was talking about for microbes, except in this case, it'll be for larger organisms. So your fish, your cephalopods, your amphibians, etc. The ecophysiology lab, so that's looking at the effect of stresses on different organisms, mostly fish, and the freshwater unit. And then where I fit in is this aquatic genomics research platform, and that's me. So I deal with everything that has DNA in it, basically. So I'm involved in all the other platforms when they want to link DNA to it, and we collaborate with researchers throughout, not just South Africa, but throughout Africa and internationally. And as was mentioned previously, we are an open access platform, and one of our mandates is if somebody needs to do genomic research and they don't have the facilities at their host institution, they are welcome to come and use our equipment. And we facilitate a lot of that kind of research as well. Okay, so that's where I'm at. We've got offices at Makanda, Grahamstown, that's where I'm based, PE, Abega, Durban, and Joburg. But I'm talking to you from good old Makanda. And I'm going to be talking specifically about environmental DNA. If I was going to talk about all the different projects that we're involved in, we would be here until midnight tonight if we were lucky, and I'm sure you guys don't want to hear that. So I've picked one small topic, which I thought would be interesting and fun to go through. And that's using environmental DNA for marine biodiversity explorations. So conventional methods of looking at specifically fish biodiversity or invertebrate biodiversity or macrofauna biodiversity is to either catch them using nets or lines or different methods, or visualizing them either through remote operated vehicles, underwater videos or divers actually noting down what they can see. But the problem with these conventional biomonitoring methods is, first of all, they're either invasive or destructive. I mean, if you're going to catch the fish, it's either going to stress them out, best case scenario, or kill them. It requires a lot of taxonomic knowledge. So if, for example, if you're a diver looking at the different fish that are around you, you need to be able to know those fish well enough to be able to differentiate them and identify them. So you need to be an expert, and there's no such thing as a person who's an expert in fish and in sponges and in crustaceans and in everything. So that becomes a limiting factor. It's also very time consuming. And one big issue is that it tends to miss the rare or shy or elusive taxa. And this is where environmental DNA comes in. It compensates for a lot of these gaps. It's not the silver bullet. It won't replace the traditional methods, but it's a very good complement because it fills in a lot of these gaps that the traditional methods do miss out on. So what is environmental DNA? Environmental DNA refers to genetic material that is shed by organisms into the surrounding environment. So this is DNA from cells, from tissues, from fluids, from poop, everything. And a fun fact is that humans shed 30 to 40,000 skin cells every single hour. So during a 24-hour, you and I lose 1 million skin cells, and that's we shedding all the way into our environment. And the same process, the same principle applies to aquatic organisms. So in this image, we're focusing on fish because it's a passion of mine. And so if we collected some water, in that water would be pieces of DNA and cells from all the different fish or other organisms, depending on what you're targeting, that live in that aquatic environment. So if you just collect the water, you're not disturbing any of the other fauna or flora that is living there, and it's not invasive, it's not killing anything or stressing anything, you're just collecting some water. So that skips out a lot of the negative impacts of traditional methods of monitoring, biomonitoring. So you collect water, usually you filter it, and then you extract the DNA. And once you've got that DNA, you can do all sorts of different things with it. You can either use a primer set to target a specific group, in this case fish, or you could target a single species, or you could target algae, or you could target microbes. And we do all of the above here at SciAb. But I'm going to focus on 2 projects. The first project is looking at the general fish biodiversity found within marine protected areas. And this project was looking at what was in a marine protected area versus what is outside of a marine protected area. So for those that don't know what marine protected areas are, those are like game reserves, but in the ocean. So they are typically set aside and they have different rules, but most of them have no fishing rules. So commercial fishing cannot happen in those areas. And the reason for that is to allow refuges for fish so that they can reproduce and grow without being fished to extinction. So they're basically game reserves. So the first project that I'm going to be talking about today is on Jessa Canyon. Now, Jessa Canyon is a beautiful marine area in the Isimangaliso marine protected area, and very little is known about the fish biodiversity in this particular canyon. So as I mentioned, it's located in the Isimangaliso marine protected area, which is around the Sodwana area. And Jessa Canyon, so these are bathymetry data, and it's 90 degrees to this image. So here would be land, and here would be the rest of the ocean. Jessa Canyon is over here, and the depth range ranges from 90 meters all the way up to greater than 580 meters. So it's quite a deep canyon. It's not the deepest we've got. Right, and white sands in the marine protected areas are deeper, but those have got additional restrictions in terms of sampling. So we wanted to start off on Jessa Canyon before moving into the larger canyons. So we sampled water in these areas. And this is just to give you an idea of the range of different fish that were detected using eDNA. Now, I've picked out a couple that are just really cool. And it's ranging from, we've detected dolphins all the way through to rays, guitarfish, sharks, etc. And many of these are actually either endangered, vulnerable, near threatened, or critically endangered. So it was a very good idea to see what's there, to provide a baseline for future management of these marine protected areas, to analyze the effect of climate change and whether the marine protected areas are actually doing their job in terms of providing refuge for these fish species, and then comparing it to outside the marine protected area. The data for them outside the marine protected area is still coming. One thing I did forget to mention, these are all ongoing projects, PhD projects that I'm mentioning here. So shifting gears a little bit. So here up until now, we've been talking about fish in general. So looking at using primers that are degenerate, which means they will bind to most fish species. And what I'd like to do now is shift to a different project where we're going to be focusing on a particular species, one species. And this species that I'm going to be talking about is very iconic, and it's very close to our heart here at SCiB because it is the reason that SCiB was initially founded. And it's kind of in our DNA, and that's the coelacanth. So now coelacanths were originally only known through the fossil record. Okay, so they were dated back to 360 million years ago, and for a very long time, everyone thought they had gone extinct about 66 million years ago. And this is an example of the fossils that people have found over the years. But then in 1938, so not so long ago, in 1938, a weird-looking fish was caught by trawl net off the coast of East London. And this fishing captain saw this weird fish, didn't know what to do with it, didn't know what it was, and he was curious enough to contact Courtney Latimer at the East London Museum, who in turn got in touch with JLB Smith. And they recognized that this live fish that had been caught was actually a live fossil. So it was around when the dinosaurs were around, and it's still around now. So it's often referred to as a living fossil, which is really, really cool. In terms of the timelines, the first one was caught off the Chilumni River, and that's why it's called Latimeria chilumni. Latimeria for Courtney Latimer, and Chilumni because that's where it was caught near the river. And then nothing happened. This one fish was caught and there was dead silence, and there were rewards put out for anyone who could catch another one. They didn't know where these populations occurred, nothing like that. And then 16 years later, a second one was caught. And this one was caught in the Comoros, and it's now known that there's quite a few individuals living in the Comores Islands. And then in 1998, a second species of coelacanths, so not Latimer chilumni, but Menadioensis, was discovered in Indonesia. And then in 2000, they found coelacanths off the coast of South Africa. So not this first coelacanth, because there was only one coelacanth specimen ever found off the coast of South Africa, up until 2000, where they found a few off the coast of South Africa. And that reinvigorated the enthusiasm for this really, really cool fish. So it's a very rare species. It's the only extant genus, so it's the only currently existing genus of the ancient Actinician fishes. There are two species, which I've already mentioned, so the African coelacanth and the Indonesian coelacanth. And what makes these very difficult to study is they're found in caves and canyons that are between 500 to 800 meters deep. So very, very, very deep. And they're listed as critically endangered because there's so few individuals. There are very, very few of these coelacanths still alive today. So some special features is each individual has a unique spot pattern, just like a fingerprint. So each of the individuals that we have discovered in South African waters, we've actually given a name to. So there's Thor, there's Beatrice, there's Mzumbi, there's Eden. All of them we've given names to because we can tell based on the pattern of their white spots which individual is which I think is kind of cool. So we actually have a little book with there's less than 100 specimens that have been identified, and each one has their own name. Their pectoral and pelvic fins move in a tetrapod-like motion, so they don't move like normal fishes' fins do. And also they've got bones, which is not like what happens in normal ray fin fishes. So even though they look like fish, they're not fish. They give birth to live young, and they have a gestation period we estimate of 1 to 5 years. There's a lot we don't know about these species still. Because they're so rare. They've got the lowest hemoglobin content of any vertebrate, and they've got very low metabolisms. Their lifespan is estimated, again, not sure, but they're estimated based on growth rings to be 80 to 100 years. And they have a rostrum, which is an electrosensory organ which helps them hunt. And what I find very cool is they've actually got a hinge in their skull that allows them to open up their jaws even wider than normal to catch their prey. And neither of these two features are found in other living vertebrates. So all of these things is what makes coelacanth particularly unique. So we know that there's a population of coelacanth, African coelacanth and Comores, all the way down to these two locations in South Africa where they've been found in canyons off the coast of South Africa. But that begs the question, why on earth was this single lone little specimen caught all the way down here in the Eastern Cape? Was it accidentally washed down here by the current? Because there's a very strong Agulhas current that runs down this eastern side of South Africa. Was it lost? Or does the population that we know exists here extend further down the coastline? And that brings us to the question, how do we find these coelacanths? Up until now, finding them has been determined by remote operated vehicle. So they send down an unmanned little sub to try and find the coelacanth, or by very super specialized divers. Because these coelacanths live at such deep depths, you need to have special gas mixers, etc., and it's extremely dangerous. In fact, some people have died trying to find the coelacanth. So they're extremely rare. So finding them using ROV or diving is very, very difficult, and most of the time you won't see them. They live at very deep depths. Not only that, but they're nocturnal, so they only move around at night. And during the day, they hide in caves and canyons. So how on earth are we going to find these? And this is where eDNA comes in. So I've spoken about using eDNA to look at fish assemblies. Now I'm going to talk about using eDNA to target a single species. In this case, we're wanting to see if we can use eDNA to find coelacanths specifically. So there's our beautiful coelacanth. This was actually taken off Mzumbe. And this pilot study we did was to take samples in Jessa Canyon here, where we know there are some coelacanths. We took water samples upstream and we took water samples downstream. And using eDNA, woohoo, we found it. You don't understand, we were super excited by this result. We get very enthusiastic. Upstream, there wasn't anything, which is expected, but at Jessa Canyon and downstream, there were coelacanths detected. Now, what we're busy doing is extending this all the way further down the coastline to see if the distribution range of the coelacanths is wider than we originally thought. So if we go back, sorry, I should have duplicated this slide. So we know they occur here, and this is where we sampled in the pilot study. Now we want to go all the way down to the coast to see if this was just this detection of the single species was because it was a stray, or was it really because the population does extend further down the South African coast. And we would also like to be able to use eDNA to start identifying which individuals, if we can find markers that can differentiate between the individuals, we want to see if we can track where individuals move. But that's a long-term goal and very much ambitious, but we're going to give it a bash. Okay, so beyond the surface, what are the potential impacts? Why are we doing this? Why are we doing eDNA in terms of marine fish research and management? First of all, improve biodiversity assessments. And SIAB, that's one of our central goals, is to look at biodiversity assessments. eDNA can detect a wide range of species, or it can detect specific species. And it's very, very good at differentiating between cryptic species. And what I mean by cryptic species, these are species that look very similar to each other, but are actually individual or separate species. So if you were just looking at them doing morphological assessment as a diver or an ROV pilot, or looking at underwater videos, you would think you only had one species. But by looking at the DNA, you can actually differentiate between these closely look-alike species. eDNA is also very, very good at finding rare or elusive or shy species, or species that are just good at camouflage. If you think about the sole, they actually hide in the sediment and they totally camouflaged. You won't see them unless they move. What's also really nice about eDNA is non-invasive. You're just taking a water sample. And once it's optimized, once you've got the pipeline in place, it's actually very, very good in terms of time efficiency and cost efficiency. What's really nice, and also another thing that we're doing, we're using eDNA to look at different invasive species, and I haven't spoken about that in this presentation, but if you want to have a separate talk about invasive species, we can definitely do that. But it's very good at detecting early detection of invasive species. When you get to the stage where you're actually seeing a lot of the species, invasive species, then it's too late to put policies in place to control them. By using eDNA, you can detect one or two individuals, and you can then get a reaction and a contingency plan put in place to deal with invasive species before they get out of hand. All of the data can then be used to inform fisheries management, marine protected areas management, and for SIAB, and particularly me, I'm passionate about my coelacanths, and it will be used to update the coelacanth management plan because they're critically endangered and they are iconic. They're part of our heritage here in South Africa. So the research I've been showing you here is mostly from Jodie Oliver's PhD, and the team involved is Kerry Singh and Stefano Mariani. and Ryan Palmer, who's the ROV pilot, and the crew of the Pakisa. Thank you very much to everybody. And of course, you've got to acknowledge everybody, including Diplomix. And to end off with, I'd like to show you a little video clip of coelacanths that we saw on our last field trip. So this one is actually called Thor because of that hammer-shaped marking on his tail. Any questions? I hope you enjoyed it.
Yes, thanks so much, Gwen. Lots of expressions on faces. We do have maybe just time for one question. If anybody wants to ask Gwen anything.
Yes, are you taking master students?
Yes, yes. We do. We have about 60 masters and PhD students and a few honor students as well at the institute. Yes.
No, but are you taking master students, new ones?
All
All the time. We're looking for enthusiastic and dedicated people. So if you're enthusiastic and are not afraid of hard work, but we've got it's a nice team. Yes.